Hello everyone, my name is Tais Labrador. I'm a senior student here at St. Thomas University. I'm majoring in chemistry and my future goal is to go to graduate school in chemistry. I did my research internship on the summer at the U.S. Department of Agriculture at the chemistry department. There I was assigned a project titled Chemical Characterization of Avocado or Persea Americana cultivars from 3 avocado races using two volatile collection techniques for leaf and bark material. So today I'm going to be talking to you about this. So the two main reasons why these kinds of projects are going on at the USDA is, first of all, because the red ambrosia beetle. The red ambrosia beetle is an insect that is threatening the population of avocados in the United States. Because this insect bores into the trees carrying with it a deadly fungus that causes the death of the tree. So what they mainly do at the USDA for this particular insect and for any other kind of insect that may be affecting the population of any tree in the United States is that they take samples of this particular trees that are being affected, of the bark and the leaf materials of these trees, and they take the samples to the lab and they chemically analyze those samples by means of a gas chromatograph or mass spectrometer that they have at the laboratory. And once they have separated and analyzed the components that are present in the sample, they pass them on to the entomologists, and in the entomology departments, they test whether these components are attractants to these types of insects that affect these cultivars. So right now, the main focus is on avocado because of the presence of this beetle. So the second project that's going to go on at the USDA in the future is the correlation between the genotypes of the plants and their chemical profiles. So we want to find out if there's any relationship between the genotypes of the avocado cultivars and their chemical profiles. So to start with my project, I want to say that there are 3 genetically distinct races of avocado. They're known as Guatemalan, Mexican, and West Indian. They're next to the bullet points on the slide, and below the bullet points you can see the 3 cultivars that I used for my particular study. Each of the 3 cultivars belong to vegetable races. So the purpose of my project was to compare the 2 methods of extraction that I used. The first method of extraction that I used was the solid phase microextraction, and from now on I'm going to call SPINI to abbreviate. And then the second method that I used was the volatile collection technique using the super Q filters. I'm going to abbreviate now, saying that that's only super Q method. And then my second purpose was to compare the chemical profiles between the cultivars of the same and different races. So at the end, I'm going to come up with a comparison of all 9 cultivars that I analyzed. So now I want to talk a little bit more about the methods of extraction and separation that I used for this particular study. So the first one, as I mentioned, was the SPINI method. The SPINI consists of a fiber that can be exposed to the sample, and that fiber can bind to the volatile chemicals in the sample, and that way it can collect the components of the sample, and those components collected by the SPINI can later be dissolved in the gas chromatograph that analyzes those chemicals by means of its molecular weight and also its interactions with the column. Then the second method that I used was the volatile collection technique using the super Q filters. The super Q filter is shown in figure A next to the SPINI. So the volatile collection technique consists of glass chambers in which the sample is placed, and then this is a push and pull system because air is pushed by a source of purified air through the glass chambers where the sample is placed, and as the air passes through the chambers, it carries with it the volatile chemicals that are in the sample, and then as air is being pulled out by the vacuum system, the components of the sample are collected on the super Q filters that are attached on this end. Then once those components are collected on the super Q filters, they are extracted using a solvent. In my case, I used methylene chloride for extraction. And then this later is injected in the gas chromatograph, which gives us the information of the components that are present in the sample. So in figure D, we can see how this information outcomes in the computer aspects. So for this particular project, this was my procedure. I basically weighed 3 grams of each of my samples of bark and leaves of vegetable cultivars that I analyzed. and then I took them to the lab, and absorption with the speedy lasted for 2 minutes. Desorption on the gas chromatograph also lasted for 2 minutes. Collection with the super Q filters lasted for 15 minutes. As I said, methylene chloride was used for extraction. For all super Q samples, a C16 internal standard was added, and then the chemicals were separated and analyzed with the gas chromatograph. For data analysis, the area percent and the Kovacs indices were calculated for all the samples. And in the case of the super Q samples, the amount in nanograms were calculated because we were able to quantify the components of the samples because of the use of a C16 internal standard. So finally, Conquest 5 and Excel 2007 were used for data analysis. So as I said before, my first purpose was to compare the two methods of extraction that I used, the speedy method and the super Q method. For this graph and for all the graphs that I'm going to show, all the data that is going to be considered are the pegs whose area percent is greater than 3%. So on the x-axis, you're going to see always the Kovacs index, because the Kovacs index, it's a standardized retention time specific to the column. That's why it was used as opposed to using the retention times. So on the y-axis, you see the area percent, and this is a comparison where the blue bars represent speedy, and the red bars represent the super Q. So as we can see from this graph, the super Q method was able to collect, at least on this cultivar, this is the leaf of the bacon cultivar, was able to collect a wider range of chemicals than speedy. Speedy was able to collect only fewer chemicals, and for some, or maybe the majority of the chemicals, like this one's right here, as the amounts that speedy collected were significantly greater than the amount that the super Q method was able to collect. This is the example of the bacon cultivar. Now let's take a look at the bark of the day cultivar. Here we see different results. First, we see that speedy as well as super Q were able to collect wider range of chemicals, and not a single method was better than the other one for collecting a wide range of chemicals. That's for the day cultivar. Now let's take a look at the leaf of this same cultivar. As we can see now, speedy was able to collect a wider range of chemicals. However, super Q in this case was able to pick up only a few chemicals. So we can see from this data how there's not only differences between the results obtained from the different cultivars, but also there's differences between the results obtained from the leaf and the bark of the same cultivar. So finally, I wanted to point out the bark of the Brooks Lake cultivar. We can see from this data how speedy was able to collect, was better at collecting the lower molecular weight chemicals. However, the super Q was better at collecting the higher molecular weight chemicals, except for these chemicals in the middle that were collected by both of the methods. And this chemical right here, that was the lowest molecular weight one, and was actually collected by the super Q. So the second thing that I wanted to point out, the second objective that I was pursuing when I did this project, was to compare all the cultivars, the chemical profiles of all these cultivars. So here's a comparison of the bark material from all cultivars using the super Q method of extraction. I'm going to show a super Q bark, and then I'm going to show a speedy bark, so you can a speedy leaf, so you can have an example of everything. So here, each Kovacs indices indicate a different chemical that was found. And right here, I wanted to point out that now we don't have anymore the area percent. We have the amount in nanograms, because this was done for the super Q sample, which had a C16 internal standard that allowed us to quantify the components of the sample. So we can see that there's a few similarities. For example, this chemical right here, Kovacs index 1396, was common for 7 out of the 9 cultivars. And we can also see a few similarities, like this chemical 1342. But for the most part, the chemicals were found on 1 cultivar, 2 or 3 cultivars. So now, let's jump to the comparison of leaf material from the cultivars analyzed with the speedy method. So here we can see a little bit more similarities between the chemical profiles of the cultivars here. This chemical at 913, this other chemicals at 965, 1345, 1396 is repeated again as a chemical in common between all the cultivars. So here we can see more similarities than in the previous graph. This was picked up by the spinning method. So finally, I wanted to point out, because I showed differences and similarities, so I wanted to show how this is the way the information outcomes in the computer. Information outcomes in the computer as fixed, this is called the gas chromatogram. So from the gas chromatogram, as far as I can see, I see more similarities actually than my data analysis told. Each color, each pig in a different color represents a different cultivar. So this is the same example, the gas chromatogram for the example that I showed, the data analysis that I showed of the bark for all super Q samples that I tested. So the patterns repeat, as you can see. So this may be showing more similarities than the data analysis that I showed there. And then the same is true. This is the chromatogram for the other, for the leaf of the spinning of all the cultivars analyzed using the spinning method of extraction. And we can see how the patterns repeat even more, and how this shows even more similarities between the cultivars. So as a conclusion, I could say that the efficiency of the methods of extraction, spinning and super Q, varied from cultivar to cultivar. We could see in the comparison between the graphs, the comparison between the two methods, how that efficiency varied from cultivar to cultivar, and sometimes from the bark and the leaf of the same cultivar. So according to the statistical analysis, especially the statistical analysis of the super Q cultivars, the chemical profiles of the cultivars were different. However, the chromatograms show more similarities between the chemical profiles of the cultivars. And why is that? I previously said that for data analysis, we consider all the pigs whose area percent was greater than 3%. And then what's only shown in the data analysis is that data that was over 3%. However, the chromatograms that I showed show everything. So a way of improving this data may be considering the pigs even the pigs whose area percent was less than 3%. And the reason why the USDA scientists came up with the idea of choosing the pigs whose area percent was over 3% is because, according to their experience and their purpose is to find out which of these components are attractants to the insects, and according to their experience, they have found that the components that are attractants to the insect are the ones whose area percent in the gas chromatogram is greater than 3%, so that's why they make these assumptions. But now they are interested also in the comparison of these different cultivars, and maybe in the future in finding the correlation between the chemical profiles and the genotypes of the plants. So maybe for the future purpose, it may be like useful or more useful for them to consider all the pigs for the purpose of comparison. So before finishing my presentation, I would like to acknowledge St. Thomas University, Dr. Pilar Mo, for giving me the opportunity of going to the USDA and having this great experience, the Science Fellows Program, the USDA, and Dr. Bob Heath for opening the doors for me of the chemistry department at the USDA. And finally, to my direct mentors, Helena Schnell and Ricardo Joseph, for their patient help and support, and to the MDCSTU Summer Research Institute for paying for the internship. Thank you very much.
Questions? Don't be shy.
Okay, questions.
Hi, the Covax. Okay, I see in some of your pictures that there's, for some particular values, even so that you have a large amount of a compound that you extracted, and when you compare that with other values of the Covax index, sometimes you have big error bars, sometimes you have small error bars, like they're sensitive. The error bars are sensitive to the index. Is there any explanation why the error bars are sensitive to the index?
Well, it's not, I don't think they're sensitive to the index. What I think is, we, as Damien explained before, we try to pick up this material that we picked up from the leaf and the barks of this cultivar, and we try to get leaves and barks that are not too young and they're not too and they're not too old also, because there may be a variation between the chemical profiles of the young leaves and the old leaves. So that's what I think accounts for the variation of the standard deviations, because that's the beauty of the biological sample. Because also the other thing that can account for the variation between the standard deviations is that there are some of these leaves and some of the barks that are being affected by insects, and some of the leaves also are actually bitten by insects. And it's believed that when a bark or a leaf is bitten by an insect, that changes also the chemical composition of the bark and the leaf. So I cannot guarantee that the leaf that I tried my best not to get a leaf that was bitten by an insect, or not to get a piece of bark that was younger than the other one, but that's, you know, I try my best, but I cannot guarantee anything. So that's what I think would account for the differences in standard deviations. I don't think it has a relationship to the Goldwasser's index.
Second, is there in your team any protocol to characterize numerically when two functions are too close to each other? Because when you're talking about the qualitative, the similarities between the chromatogram, between different results, they're very close to each other.
Yes.
You're doing that on the qualitative, based on the qualitative point of view, just by looking at the graph. But is there in your team any, I would say, protocol, mathematical protocol, that might tell you exactly the degree of closeness between these two functions? Is this or that?
Let me first make sure that I understood the question. You mean this, right? That this peg is really close to the other one, right?
Yeah. Suppose that you have the blue graph over there, the blue function.
Okay.
And then you have the other color. I don't know if that's brown
The red color.
or red color. So if you want to say the behavior of these two functions are too close to each other, is there any index that you can use to characterize how close these two functions are?
The index that characterizes how close these two functions are is the Goldwasser's index, actually. If they have this cultivar for this pig, for the Goldwasser's index, what we use is the retention time. So according to this retention time, we normalize this retention time to the Goldwasser's index. So if this pig that is at 2.6 in retention time, the computer can sense exactly and can say which one is at which retention time. If this pig, let's say, that is at 2.6 in retention time, and this other one is at 2.6 retention time, we can normalize that. And if the Goldwasser's index is the same, it's believed that it's the same substance. It's believed that it's the same. We don't know. This is only a qualitative analysis because the gas chromatograms give only qualitative data. The Goldwasser's index cannot say this is the same compound or this is this particular compound. What says what is the mass spectrometer, which is the other technique that they use at the lab, but I didn't have the opportunity to use. The only one that I used was this qualitative technique. Okay. Right, but well, theoretically all the leaves contain the same components, and all bars contain the same components. Well, but we needed like the thing is that when we put the samples, we have to expose those samples to a spinning, which is a fiber that picks up the components on the environment. So we need a significantly greater or great amount of leaf or bark in order for the spinning to pick up those volatile chemicals. So we need, that's why they come up with this idea, 3 grams, and I cannot get 3 grams from one leaf, but I understand your point, yes. That may account for the standard deviations, yes.
Last question, I'm going to take the honor. Did you ever consider screening for pollutants? For instance, if the leaves were gathered close to a highway, we're talking about pollutants from the car, we're talking about pesticides, herbicides, because that might influence the student. Did they ever consider testing the quality before they do anything else?
No, not that I know of. They didn't test. This is just a preliminary analysis. They didn't test anything else. And I just went out, grabbed the leaves, took them to the lab without any preliminary analysis. But I know they have done this analysis before, but I do not think they have tested anything before doing any other particular parameter like pollutants or anything else.
Because we're crowded with cars around Miami. Okay, thank you.